All right, so we are recording. So I'm really excited to continue our variant type series today and honored to welcome Dr. Chimene Kesserwan, who is doing a clinical hematopathology fellowship at NYU and also works for the NCI-NIH. So really excited to have you here today to talk to us about matched tumor normal sequencing. So I'll let you take it away.
Thank you, Beth, and thank you for the invitation. And hello, everyone from New York City. So today I will be talking about the curation and interpretation of sequence alterations in cancer, a tale of two genomes. Feel free to interrupt me if you have any questions. So we'll try to make it as interactive as possible. So I only have one objective. I will demonstrate how the integration of matched tumor normal data could inform the classification and the interpretation of germline variants. And we'll go through a few examples to demonstrate the process. But first, I would like to set the stage by reviewing how the field of cancer genomics has evolved. We know that the Cancer Genome Atlas and the International Cancer Genome Consortium contributed significantly to our understanding in cancer. And this is an adult, right, in the adult space. In the pediatric space, the landscape of cancer was unfolded via the effort of the Pediatric Cancer Genome Project that is led by St. Jude, and in addition to the wealth of data that we have via the target database. So, but I would like to pause for a second and talk about PCGP. This Cancer Genome Project was led by Jim Downing from St. Jude and Elaine Mardy, then at Washington University of St. Louis, now at Nationwide in Columbus, Ohio. So this project, which launched in 2010 and ended in 2013, what they did is that they did whole genome sequencing on 700 tumor normal pairs and whole exome RNA sequencing on 1,200 sample. And the Pediatric Cancer Project expanded our knowledge of the molecular underpinning of childhood cancer and led to a deluge of high impact publication unfolding the landscape of pediatric cancer, and has also marked a new debut for cancer predisposition in children. So here, most of you are familiar with this paper. Is a landmark paper by the St. Jude team by Jinwei Zhang, Michael Walsh, Gang Wu, and other authors where they looked at cancer predisposition in children, and they did find that 8.5% of children with cancer harbor a germline predisposition. What they did is that they looked at some 565 genes in the germline. Of these 565, 60 are associated with autosomal dominant cancer predisposition syndromes listed here. And here you see in this two-dimensional plot here, we see that we have pathogenic and likely pathogenic variant across various cancer predisposing gene and across various cancer types. You notice probably that the TP53 is the dominant cancer predisposition gene in that paper, that there was some ascertainment bias because St. Jude is known for its rich population in adrenocortical tumors, adrenocortical carcinomas, specifically from Brazil. And so we have variant, we have Liframeni syndrome caused by the German P53, and this patient was adrenocortical tumor. But at the same time, we found additional cancer predisposition variants in the non-cancer predisposing gene. So as we mentioned, the PCGP and PCGA and other large data expedited our understanding of the cancer genetics and genomic for in pediatrics and in adult. And here is a list of the first pilot studies of matched tumor normal in pediatrics oncology. This list is from the US, but as well we had so many other studies performed across Europe and in Australia. So the first one, Pete's My OncSeq, is from Ann Arbor, where they studied 102 patients with solid brain and liquid tumor. They did whole exome and RNA-seq, and they detected like 46% potentially actionable findings in terms of therapeutic, prognostic, or diagnostic actionability. But they did come up with 10% of their cohort had a cancer predisposition syndrome, very pathogenic, likely pathogenic variant in one of the cancer predisposition gene. The VICC from Baylor, Bill Parson and Sharon Plon, they did find in their cohort of solid tumor, the rate of germline finding was 10%. The clinomic from NCI, led by Javed Khan, they did find 12% germline findings, and the PEP-seq from New York, from Columbia, this number was 14%. Now, meanwhile, at St. Jude, in leveraging on the PCGP findings, the Genome for Kids protocol was launched in 2015 and led by Kim Nichols. She is a international expert in cancer predisposition syndrome and in leukemia. So Kim was hired in 2014 to St. Jude from SHOP, and she hired me in 2015, where I worked closely along with other colleagues on this Genome for Kids protocol, where every child admitted to St. Jude will have the option to have his tumor sequenced via whole genome, whole exome, and RNA-seq, and a paired matched normal sample will also receive whole genome and whole exome sequencing. And in the tumor genome, we'll be looking at single nucleotide variant, indel, copy number variant, looking at tumor ploidy and fusion transcript. And in the germline genome, we'll look at single nucleotide variant, indel, copy number variant, and DNA ploidy. So the primary objective of the G4K was to identify and characterize actionable alteration in paired tumor normal samples. So we have enrolled 309.
And I have a quick question for you, actually.
Sure.
If you go back to the previous slide, when you're measuring DNA ploidy in the germline, is that primarily to rule out contamination?
Yeah, constitutional.
Okay.
Constitutional, but for the ploidy, like for hypodiploidy, we use the tumor sample.
Got it. Yeah, I was thinking because germline aneuploidy is relatively rare and usually like very well known, but
right. We have a few samples of like few patients with trisomy 21, not trisomy 21, with megakaryocytic leukemia. So, but outside we did not have any finding. That is a good point.
Okay, cool. Thank you.
Sure. And feel free, please, to interrupt me for any question. So we have enrolled 309 patients, newly diagnosed 85 and relapse refractory 15%. mostly hematologic malignancy, because St. Jude is quite expert in heme malignancy, but as well with CNS tumor and solid tumor. And out of these CO9, 300 received germline analysis, and 253 received tumor analysis. And here also is a breakdown of the various tumors. So what did we find? And this, the findings were published recently in Cancer Discovery. So we had 58 variants in 29 genes in 55 patients. So our germline findings, 18% of patients carried at least one pathogenic or likely pathogenic variant. So if you take the 58 variants that are pathogenic, likely pathogenic, 25 of the 58 are in genes relevant to the patient tumor type. What do I mean by this? Meaning RB1 in retinoblastoma, NF1 in pilocytic astrocytoma, right? P53 in adrenocortical carcinoma, and so on. But we found 33 variants in genes with no relevance or uncertain relevance to the patient's tumor type, and or variants associated with adult onset cancer or autosomal recessive cancer predisposing syndromes. Let's give the example. For example, we found a BRCA2, what is the BRCA2 here, in a CNS, in a child with glioma. So we did not know an association. It's not one of the core tumors in the BRCA2, and it happened in a child. So this is one example. So is that an incidental finding, or is it relevant for the tumor in any way? We did find WT1 in leukemia. So, and we did find SMARCA4. Where is SMARCA4 here? In a case of neuroblastoma, we know SMARCA4 is associated with atypical teratoid rhabdoid tumor, but we do not know of an association with rhabdoid tumor predisposition syndrome. So is that an incidental, or is it causal in any way, but yet we do not know the association? And in fact, after we reported this patient, we did find that the team, Dr. Plum team from Baylor, has reported another patient with SMARCA4 in neuroblastoma. So we think there is a new association between SMARCA4 and neuroblastoma. So in addition, we did find the variant of uncertain significance. So 77% of the patients had at least one variant of uncertain significance. And that was a meticulous work where we did go over each one of these variants and follow the ACMG guideline for classification. But the first question that we asked, can we use tumor data to better inform the classification of germline VUS? And the second question, can we use tumor data to better inform the relevance of germline PLP variants in relation to the patient's tumor type? Is it incidental or causal or relevant to the tumor? So these two questions were in our mind when we had the data, and we tried to come up with some association. But I want to here mention, we all are familiar with the ACMG Richard et al. Paper on the germline, on the rules, guidelines, recommendation for germline variant curation, and we all are following this guideline. And recently, which I did not put, I meant to put a slide for it. We know that recently, the VICC group, along with the Cancer Genome Consortium, they did come up with the oncogenicity classification geared towards classification of somatic variant, similar to the Richard et al. Paper for the creation of germline variant. However, the ACMG and guideline, the Richard et al., did not address how to integrate evidence generated by tumor data when we assess the pathogenicity of germline variant. Therefore, the ClinGen established a multidisciplinary working group, the Germline Somatic Variant Subcommittee, with this specific focus in mind. As shown here in this, and here are the leader of this group, and you all are familiar with them: Sharon Plum, Dimitri Sonkin, Michael Walsh, and Deb Ritter. So, like you see in this graph, and after reviewing meticulously and analyzing available resources and performing so many somatic integration curation exercises, it was determined that somatic hotspot could be systematically integrated into moderate evidence of pathogenicity. Here, labeled in green, it was given the green light by the ClinGen leadership. So we can apply now the PM1 for somatic hotspot, and I will tell you shortly how do we determine if a variant is a somatic hotspot. And I want to make sure that we all are on the same page. When we talk about hotspot, we mean that it's a recurrent mutation variant that is driving the cancer. So the second somatic data element that could inform germline curation is RNA-seq. If RNA-seq is indicative of a splicing alteration, this may be considered a strong evidence in support of germline pathogenicity. And other somatic evidence, such as tumor mutational signature, could be considered supporting evidence of pathogenicity. However, these evidence codes labeled here in yellow should only be assigned under guidance of a multidisciplinary tumor board. Also, this working group determined that at present, an evidence to support pathogenicity, to integrate the loss of heterozygosity as an evidence for pathogenicity of germline variants would require some additional caution. And again, it needs to be determined under the guidance of a multidisciplinary tumor board. So you see, we have color-coded the green light, which got approval from the ClinGen leadership, the yellow, like you need to be careful, but now we have the red for the loss of heterozygosity, but yet it is up in the provider discretion and after the consensus of a multidisciplinary tumor board to apply these codes, and we have applied these codes in our study, and I will walk you through example. So this is what we addressed in this somatic germline task force, which continue its effort to further develop and update this rule, but yet one question remains to be addressed: how tumor data may help to refine the interpretation, meaning the relevance of germline variant relative to the patient's tumor in question. So here you have, like I mentioned, assume you have a germline pathogenic or likely pathogenic variant, right? It could be relevant to the tumor type. So, for example, TP53 pathogenic variant in a child with adrenocortical carcinoma. We know this is a causal variant relevant to the tumor type of this patient. But we have some variant, as I showed in prior slide, of uncertain relevance to the tumor type. So either the association is not previously reported, and I gave the example of the SMARCA4, which we found in a patient with neuroblastoma, or the association is with tumors of childhood onset not well established, like the BRCA2 pathogenic variant in a patient with glioma. So now I will walk you through 3 examples. If we don't have time, we'll go only over 2 examples where integration of tumor data helped refining the classification and interpretation of variants. And all these examples and additional examples are all included in the Genome 4K paper. So, but first, what type of tumor data we have interrogated? We looked at hotspot mutations, and you all are familiar probably with the cancer hotspots, which is by Barry Taylor group, by the MSK group. And this is our reference for the mutational hotspot, but we acknowledge the as well as Horag et al. In their paper acknowledge that the Cosmic database is another also database for hotspot, but mind you that the hotspot in the Taylor et al. Paper and at this website, they took into consideration not only how often the variant is seen in a specific tumor type, the recurrence, how often it is recurrent, but they devised quite a sophisticated statistical method, which is beyond my expertise nor understanding, where they looked at the length of the gene, the mutation rate. So based on all these together, not only how often do we see this variant recurrent in a specific codon. So the second data to integrate, like we talked about, the loss of heterozygosity or second hit. Then the tumor mutation burden, microsatellite instability, if you have a high tumor mutation burden and microsatellite unstable tumor, and you have a variant of uncertain significance in a mismatch repair gene, in the appropriate context, that may give a supporting evidence of pathogenicity. But I think now there is a VCC for the Lynch syndrome and mismatch repair that will put together the specific guidelines for how to make use of the tumor mutation burden and MSI. And the RNA-seq data, which really is quite useful data to look at splice alteration, in a sense, if you see exon skipping or intron retention, that would be in support of a variant being pathogenic. And again, I'm using the word support at all time. I'm not saying it will establish a pathogenicity, because as we all know, we need to have additional evidence. It's not only based on one level of evidence. We need to compile various levels of evidence. And the mutational signature, yet it is not, it is at a research level. It did not make it to the clinic yet, but that would be also as well another molecular mutational, another data that we can, that may help us, give us an idea about the fingerprint of the variant on a tumor, which, if I have the opportunity to come to show you the last case, will show you how we integrate the mutational signature. Now we'll start with our first case. So we finished the introduction. Everything I talked about is to start now the exercise. The first case is a patient, 5-year-old with neuroblastoma, and he was enrolled on G4K. We did per tumor normal next generation sequencing testing. We did find in his germline, and the matched in his germline, we did find a TP53 variant, A161T C dot for 81G2A. So the so this is the protein paint of the P53, and color coded are the various domain. So this patient received both, as I mentioned, tumor and germline. This is the germline finding. Let's start with the germline finding before I tell you about the somatic results. But before that, location. Location is very important. Here is again from PCAN St. Jude. I pulled all the pathogenic and likely pathogenic variant of TP53 from ClinVar. In red are the pathogenic, in yellow orangey is likely pathogenic. But mind you, you may need to sometimes check for accuracy, because sometimes you may be missing some variants. So it is an excellent website that is my to-go website every day. And I am lucky to have worked on this website and use this data for during my position at St. Jude for more than 4 years. So back to our variant. Our variant is here. So you can pull your variant and you place it through this website. This is the variant here, and it is in the DNA binding domain, right? So now let's start with curation based only on the germline. We follow the Richard et al. Paper, population data. We looked at nomad, and it is absent from nomad population, and so we gave it BN2 supporting. We know that the BN2, the FinGen has recommended to downgrade the moderate to supporting level for the population data. Then the functional data, it is beyond the scope of this talk to go over how I came with PS3 moderate, but I want to bring to your attention the new TP53 database that has migrated from the IARC to NCI. And so the data now is maintained by the NCI by Sharon Savage and team. And I am fortunate to sit on the somatic task force of this TP53 database. I co-chair along with Pierre Hénon from France, who is original who is the expert in the TP53 who started the IARC TP53 database. So now it is maintained by NCI, and we have published in a small paper by Calvin and De Andrade, who is a fantastic scientists that work with Dr. Savage group, and he led all this effort for the transition. And you can find the transition paper in PubMed. So, but what I wanted to also mention that we have, as you are aware of, the variant creation expert panel for P53, where rules for P53 germline variant creation were specified, specific, like we don't say modified, we say specific rule specification based on the characteristic of the P53 gene and the phenotype associated with the gene. So, and also this, the rule specifications are published, and we follow specific rules for functional data weight assignment, including the transactivation activity, whether a variant is dominant negative or it has loss of function, and we use well curated publication for this. So according to this VICC group, we created this, we gave a PS3 moderate for this variant, and in silico data, according as well to the VICC rules classification, it received a supporting evidence. We have now PN2 supporting, PS3 moderate, and PP3. If we import all this to the ClinGen variant creation interface, it will give us an uncertain significance. So we have here one moderate and two supporting. So this variant is called as uncertain significance. So is that enough? Are we satisfied with the VUS? Do we call it VUS and go on to the next patient? In fact, we said, no, we need to investigate further. So what data we need to include? Let's look at the somatic results. This patient somatic result showed a single nucleotide variant in ALK, is a missense variant in ALK. We know that the ALK are driver for neuroblastoma, right? But would this variant help us to refine the classification of this VUS? No, it will not. And he had gains of several chromosomes. So the somatic results on this patient-specific somatic result did not help us to further refine this variant. So, okay, I showed you one example where somatic result did not help us, but did we stop? No, because now we need to look at not at a patient level, at a database level. Is this variant a germline hotspot? And so here are the germline hotspot in P53. I think I'm missing one here, and this image is courtesy, I forgot to, here, it's courtesy of Emilia Pinto from St. Jude. So our next question, is this variant a somatic hotspot? For that, we go to cancerhotspot.org and we pull the P53A161. We're looking at the codon level, right? And for the alternative amino acid T, it was reported in 12 samples. And so it is a hotspot codon. Can we use this information to help reclassify this variant? Let's see. We go to the rules and regulation set by our somatic germline committee. So is it a germline hotspot? It is not. If it was a germline hotspot, assume we have the 337, the arginine 337H codon, the Brazilian founder mutation, it is already a hotspot. We know we use PM1 for it. We don't look at the somatic hotspot because even if you found it in the somatic, it's not somatic. It's a germline. This founder mutation, the R337H. So now the other question, is it a somatic hotspot? So if yes, we can apply PM1 if we have greater than 10 samples at the hotspot residue. So, and we did, I did show you that we have 12 sample at this hotspot residue for the T, so we applied the PM1 for this variant. So now we apply PM1 and the VSEP, the P53 VSEP, they have adopted this rule as well. It can be applied to variant in hotspot 75 to 45 to 48 to 49 to 73 and to 82, but not to variant within functional domain. And so we applied this rule, we integrated this rule, and now what do we have? Was the addition of other variant. Now it is a likely pathogenic variant, and that is important because having a likely pathogenic variant in a P53 have implication not only for the patient, but also for the family. You need to do cascade testing for at-risk relative and institute the appropriate surveillance. And by the way, this variant also, I did run this variant by Sharon Savage and Megan Fron and her team, and we have a consensus that that is a likely pathogenic variant. So that was the first example where I showed you how to integrate the data, general data from the hotspot. Let's move to the second example. A 10-year-old male with BALL and cafe au lait macules. Here's our patient. He has BALL. Notice here, our tumor is here the bone marrow, but our germline is not the peripheral blood, it's skin fibroblast culture because we are looking for true germline, right? So the germline revealed a NF1 variant, C.691 plus 3A to G. So this is a splice donor at position plus 3. It's not a canonical splice. We know canonical splice are +1, +2, -1, -2 for acceptor, +1, +2 for donor. So it is not, but it's close enough. And so we need to be careful and we need to start to look at evidence. How do we call this variant and what evidence do we have? So here, just I want to bring to your attention that make sure you are looking at, if you have a transcript, you don't find it, look at the canonical transcript, at the alternate transcript. Sometimes it's mentioned in one way or the other way. And as you know, the Gen recommend you use the main select transcript at the canonical transcript. Now for the somatic, let's see now here, before going to the somatic, let's do the germline variant creation before integrating the somatic data. Population data, it is absent or very rare, I can't remember, but it has satisfied the PM2 supporting. And in silico predictive data, so we use Splice AI, which, as you know, is a publicly available splice alteration tool based on artificial intelligence, and they devised a really very sophisticated model, neural network to come up with the prediction, but still it is a prediction tool, right? And so I put in this the genomic coordinate for this variant, and it gave me it probably causing donor loss. But here I bring to your attention that so for this variant, for this NF1, we do not have yet a variant creation expert panel. I think it is in the process, or it has just formed. I'm not sure. So we do not know which threshold to apply. But here we felt comfortable to apply the PP3 again. That is important. We need to follow the rule when they exist. If they do not, we go back to the Richard et al. And but we need to look at the big picture and put into the context, and it's at the provider discretion after he studies the case, meditate on the case really well, and in consultation with other team member to come up to decide, yes, I want to give it PP3. When you feel comfortable, when you give a variant a code and you are comfortable with it and you are following the rule, you go for it. Otherwise, you say, I don't know. So now we put these two codes, and the PP3 and the PM2 supporting, and we came up again with insufficient evidence and variant of uncertain significance. Now we will use the tumor data on this patient. So we have on west of his bone marrow blast, we have NF1 intragenic deletion. So it's a focal loss of heterozygosity. You remember I told that loss of heterozygosity still, because we did not yet come up with very defined rule. We did not validate this the focal loss of heterozygosity. So it's in red, and but this is a second hit. So we know this patient already has a second hit. And we looked at RNA-seq data, and here is the gene. These are the exon. Our variant is in tronic region here, and the RNA-seq showed, you see the histogram here, showed that some read has kept the 45 and went directly to 46. We have some read that still are available between as they should be from 45 to 46, but we have some exon skipping. So we have an indication of an altered splice. So we have splice alteration, and we felt comfortable to give it PVS1, and did not consider the focal LOH even. So with that, we gave it PVS1, and now the variant is likely pathogenic. We consulted with Bruce Korf lab, and they also indicated to us that they have seen this variant in a setting of NF1. So the final classification is likely pathogenic. So now we will go to our last case, and we will have lots of time for questions. So this is a 5-year-old with unilateral retinoblastoma. On the germline, we found two pathogenic variants. The first one is RB1. Noticed it's mosaic at 8%, and that's why probably he developed unilateral retinoblastoma at an older age. We don't know. But at the same time, we got a mut-YH at 46% variant allele fraction. So this is a mut-YH, and we know that mut-YH like biallelic mutations, pathogenic mutations in MUT-YH will be associated with MUT-YH associated polyposis, not the monoallelic. So knowing that we are seeing this recurrent variant oftentimes, and when we do like a comprehensive germline panel, and we do not know what we think it is incidental. It's the patient is a carrier rather than it is contributing to the tumor. But our information is still evolving. But since we have tumor data, we wanted to see if we can integrate any of our tumor data to better refine the relevance of this variant. We know it's pathogenic, but we want to see, did it contribute to RB1 and to retinoblastoma? We do not have such association in the literature, and we know that the patient has RB1, although it is a mosaic one. So in the somatic, we did find the second hit. Yes, we did find a second hit in RB1. So we have the first one and we have the second one. But also for MUT-YH, we did find copy-neutral loss of heterozygosity. So we have two hits also in MUT-YH. Now we looked at the mutational signature, and it showed us a mutational signature 18, which is indicative of reactive oxygen species signature, ROS signature 18, which is beyond the scope of the talk to talk about the mutational signature, but here how it's relevant to the patient's tumor. So mutational signature from whole genome sequencing of this patient shows that 60% of mutations in the tumor are attributable to damage by reactive oxygen species. So let me show it to you here. You see, this is our patient. We have the various mutation signature, including the clock one, the age, which is we'll see across the board, the POL-E mismatch repair, the tobacco, MUT-YH, ROS here, and unknown. So our patient has a signature for ROS, right? And these are all the retinoblastoma patients in our cohort. So we compared it to the other retinoblastoma in our cohort, which did not display any similar mutational signature. But again, this is in the realm of research. This is not ready for the clinic. So we did not make any assumption based on it. And again, this variant is pathogenic. The way we counsel the patient is based on the fact that this is a monoallelic, the patient is a carrier. And if they ask us, did this variant contributed to the retinoblastoma, we tell them it's not reported in the literature. We do not know at this time, but we have this observation here. Another observation, he has a mutational burden that is quite high. Here is our mutational burden in the upper quartile compared with other retinoblastoma patients. So this variant glycine 396 aspartic acid is thought to be likely relevant to the patient retinoblastoma in addition to RB1. If you would like to have additional information, it's also present in the published paper. And this is a quite basic table. I will not go over it, but I will mention this table is included in the paper where we have included all the variant, and we have looked at various questions, whether the germline testing is indicated or not, what is the germline genotype, whether we have or not second hit, the molecular phenotype, and whether it's disease related. The stars are the one that are seen in a novel context, like an ATM in a high-grade glioma, FANG-G in a melanoma, SMART-A4 in neuroblastoma, the P53 in ganglion neuroblastoma. In fact, the patient, I need to make a correction. Our patient had a ganglion neuroblastoma, not the neuroblastoma. I mean, it's a well-differentiated tumor, but it's part of the spectrum. and you have the table. You can revisit this table at your convenience. So our take-home point is that simultaneous assessment of tumor and germline genomes hotspot mutation can improve variant classification. So this is what what is recommended to use at this time. But however, at present, other somatic data are not recommended to be systematically integrated. Tumor RNA-seq and the tumor phenotype should only be considered under guidance of multidisciplinary tumor board, and this requires patient-specific integrated data and institutional norms. Additional caution needed with LOH to ensure focal loss and relevant germline variant on retained allele. With that, thank you all, and I would like to acknowledge the G4K team at St. Jude, specifically my previous boss and my friend Kim Nickel, and so many other people who contributed to this wonderful work. And of course, I would like to acknowledge the ClinGen somatic germline working group, which I am part of since 2016, I think. So with that, thank you so much, and I am happy to answer any question.
Great, thank you so much for that. I mean, so many of us are kind of trained to think of germline somatic paired testing as germline subtraction only, and I think these data really show that you can leverage the somatic data to interpret the germline as well. So that's really cool, and something that we should think about automating in the future. I think it'd be really cool. So yeah, if anybody has any questions, go ahead and jump in.
I would like to ask a question.
Sure.
Hi, Chimene. Congratulations. That's a great talk. I learned a lot. I have two questions for you about the case you showed with the NF1 splicing mutation.
Yeah.
I just wonder if I don't know if you have germline RNA data. If that, do you have?
Yeah, we did not run. This is a great question. We did run on tumor only. We are not running. That is a good point. And we have been asked this question about RNA-seq, specifically on the blood, for example, specifically that we did run it on the tumor, but we did not do it. Now, but we did not do it. I don't know why, honestly, but in general, we know now that like various companies are starting to do the RNA-seq on the peripheral blood to help refine the classification of germline variant. But again, we always have this question of whether it is the right tissue, whether it is expressed or not at the level of germline. What do you think?
Yeah. Well, I was just curious if the exon skipping was there too, right? And then we could have this information, maybe even not having the tumor.
Yeah, we do not do RNA-seq on blood.
Right. And the other point, do you have an objective way of measuring the exon skipping? Just you showed us, it was very clear, but some cases are not that clear.
So if you look at the IGV, and that shows the sashimi plot. On IGV, the sashimi plot is more easy to navigate. It will give you really exactly the number of reads, and it's not my area of expertise. I will defer to the computational people online to comment, but myself, being naive in the bioinformatics, what I do, I look how many reads are skipped to determine, and I think in the paper, they did mention exactly the number of reads that are skipped.
Right. Okay. Yeah, we're having trouble because sometimes it's not very clear what the cutoffs are, right? So what would be considered normal for a gene, since we have a lot of alternative splicing going on in our tissue.
And we may not be able to rule out leaky splice. And what do we do in case we have a leaky splice? What would be the out effect? And so these are all evolving questions, and so hopefully we will get to learn more about this.
Yeah, but congratulations, great cases.
Thank you.
Fantastic. Yeah, thank you, Paulo, for those questions. Those are great.
Sure.
So I have one kind of simple question. When you were doing the germline testing, did you find that there was a preferred sample type for that? I mean, I know you saw blood and tissue.
Yeah.
Is there one that's preferred over the other for you? I know that blood is easier.
We used only peripheral blood. It was validated. The platform was validated for peripheral blood. I do not know now if they have validated the skin fibroblast culture. When I was there, we used to send out if we had a skin fibroblast culture because we used only the peripheral blood. For the leukemia, we used to do the skin fibroblast culture.
That sounds good. I've even heard of people, you know, kind of venturing into trying to use cheek swabs instead of.
Yes. So at NIH, we have validated the platform that is currently running. We validated on saliva. We are doing tumor normal at NCI in the laboratory of pathology on peripheral blood and on saliva, I mean. Now we have validated the cheek swab. We have some groups at the NCI that use even the fingernails, but the platform, bless you, the platform at NCI is not validated for the fingernails yet. And you wonder if someone scratched somebody else and you get the fingernail, who's
yeah, that's
just collecting.
Right. That's just such good news for our patients. To not have to go through such invasive procedures for a germline sample. So awesome, great work. That's so exciting.
Sure.
Any other questions or comments?
Great, we can give 5 minutes back to everyone's day.
Yeah.
But I'm available at any time. Feel free to send me email. I see some question in this chat.
Oh.
No, just okay.
Just comments.
Yes.
I did leave a comment in the chat. I saw that you use protein paint as a visualization.
Absolutely.
I use it frequently as well. It's a really nice tool for being able to visualize your own data.
Absolutely. It's such a great tool. And also the Genome Paint is another tool from St. Jude. So the protein paint is, and the Genome Paint, if you go genomepaint.org St. Jude, also it's a very nice tool.
Great. Thank you. I know I gave a short tutorial kind of live for those who joined the Civic hackathon in St. Louis before the CGC meeting, but I'd be happy to do that or pull somebody in from St. Jude's that maybe has some inside knowledge on development or something too. Yeah, so thank you. It was cool to see that. All right. Great. Well, thank you so much again. This was a fantastic presentation. And we do have a question in the chat.
So you would like to answer? Feel free to answer.
Yep. So Dr. Song says, does the protein paint cover mutations in adult cancer? Well, there are cosmic mutations. So mutations that are in cosmic or in civic will also show up in protein paint just as a separate track. And additionally, if you have mutations from your own data set that you would like to see visualized with that construct, there is a way to do that as well. Yeah, I don't know if you have anything to add.
No, and you can download all these data, the cosmic data, the Canvar data, and your data into an Excel spreadsheet to manipulate further.
Great. Great. Awesome.
Thank you for the invitation again.
Yeah, you bet. Thank you so much for being here and sharing your expertise with us. It's refreshing to see somatic data informing germline data. It's just really, it's exciting for our field, and we're really grateful for all the work you and your team have done. So with that, I'll end the recording. Thank you all for joining, and I'll make the recording available as soon as I can.
Thank you.
Thank you so much. Have a great day, everyone.
Bye.